start with urinary physiology, so looking at the details about the production of urine as our blood is being filtered in the kidneys. So this is kind of that blown out picture of a nephron, and our blood is going to flow into the nephron coming through an afferent arteriole. It's going to be filtered in this glomerulus, which are these specialized fenestrated capillaries. So glomerular filtration is the first process in urine formation. And what is going to happen with that is the passive process, and hydrostatic pressure is going to push all of the fluid that's in the blood through these fenestrated capillaries, and they're going to catch in the capsular space there. So the only things that won't be pushed out of blood will be large proteins and cells. So the fluid then that's going to collect in this capsular space is called filtrate, and it's at equilibrium with blood plasma, or I guess chemical equilibrium with blood plasma, with the exception that our blood flowing through here is still going to have our blood cells and proteins in it. So then it will exit the glomerulus in the efferent arteriole and will then circulate in either peritubular capillaries or vasorectal capillaries. So when you're thinking about urine production and what's going on in the kidneys, you have to track the movement of two fluids, blood and what's going to become urine. So after this filtrate leaves the glomerular capsule, it's going to move into the proximal convoluted tubule, and that's where we'll have our next two processes begin. Tubular reabsorption, which is the movement of useful substances from the tubular fluid back into the blood. So tubular reabsorption would be bringing things that we want out of that fluid and pulling it back into the body. And then the last process is tubular secretion. This also begins in the proximal convoluted tubule. And what happens in tubular secretion is that we take things out of the blood that we don't want in the blood and are going to add it to the fluid passing through here so that it can be concentrated in urine and removed from the body. So the fluid then passing through our whole tubule, our whole renal tubule, we'll call tubular fluid. And tubular fluid is different from filtrate because we've started pulling things out of it and putting things into it. So it's going to be different. It's still not urine because all throughout the nephron loop and distal convoluting tubule and collecting duct, we're going to be pulling things back out of it. So it's not until we get through the collecting duct down to these little tiny ducts called papillary ducts that we get urine. So all of the fluid passing through here would be our tubular fluid. And when we get to the collecting duct, the major job of the collecting duct is to reclaim a lot of water, to concentrate that tubular fluid so that it becomes urine. Yes?
What did you say that second one in the green area was called?
This is the nephron loop. Tubular fluid still. Yep.
Okay.
So as soon as we enter the proximal convoluted tubule, it will become tubular fluid. It's going to remain tubular fluid as we move through the nephron loop. Through the distal convoluted tubule, it's still tubular fluid. Still tubular fluid throughout the collecting duct. It doesn't become urine until the papillary duct when we can't do anything else to it. We've made all the adjustments to it that we can. So today we're going to talk about these three processes in greater detail. So glomerular filtration happens in that glomerulus, or in the renal corpuscle. And we've got two structures in the renal corpuscle that are going to be involved. So the outer part of the glomerular capsule is solid and the cells are held together by tight junctions so that the filtrate won't leak out. But the inner layer is, the visceral layer, has these specialized cells called podocytes that have these little feet that are going to wrap around. So this outer layer would be our parietal layer of the glomerular capsule. This is also known as Bowman's capsule, so you can, either name is acceptable. The inner layer is the visceral layer, and the cells there are called podocytes. The podocytes wrap out these perivascular feet that are going to wrap around our arterioles that are coming through. This picture is not really doing it justice, but our afferent arteriole, remember, is coming in, and then we have this tuft of arterioles passing through called the glomerulus. And so these would be our glomerular capillaries, and they're fenestrated. And if we look at this inner layer, then filtration is going to happen across what's called the filtration membrane. And the filtration membrane has three parts. The simple squamous epithelium of this fenestrated capillary. So we'll say that this is our filtration membrane. This is the surface across which filtration happens. And it's got three parts. One, our simple squamous epithelium of our glomerular capillaries. Two is coming from these podocytes. So we'll say the podocytes of the visceral layer of the capsule. And then three, the basement membrane that they share. So these podocytes wrap these little pedicles, little mini feet, around our arterioles and create what are called filtration slits. So the filtration slits are going to control the movement of materials so that big things aren't getting out of these fenestrated capillaries. So our fenestrated capillaries have holes in them, which makes them very permeable, and these filtration slits are going to make sure that not everything is getting out of the blood. So most everything is getting out of the blood, but proteins aren't getting out and cells aren't getting out. When thinking about what's going on with filtration, the most important pressure is the hydrostatic pressure on the blood coming through the glomerulus. But there are other pressures you could imagine. So we've got our afferent arteriole, which has a wider diameter than our efferent arteriole, and that's going to create the biggest pressure gradient in the system. So it's going to create what's our hydrostatic pressure on the glomerular fluid to move out and into the filtrate. But you can imagine hydrostatic pressure is the fluid, or is the pressure that any fluid exerts on a membrane. So once we have filtrate in here, it can exert a pressure back on the capillaries. So that's one pressure that's acting kind of against filtration, is the hydrostatic pressure from the filtrate. But then also, as we're squeezing all of our water and dissolved solutes out, then there's going to be some colloid osmotic pressure pushing or kind of drawing fluid, wanting to draw fluid back in. So overall, when looking at what's going on in the system, we have to calculate what's called net filtration pressure. And you look at that by taking your glomerular hydrostatic pressure. So it would be your glomerular hydrostatic pressure. And then you subtract your blood, the colloid osmotic pressure that is the pull of water basically from the filtrate back into the blood, and your capsular hydrostatic pressure, that is the push of the fluid back into the capsule. So we have to take all of these things into consideration to decide how much filtration is actually going to happen. So we've got our blood osmotic pressure. And so this total value will give us what's called our net filtration pressure, or NFP. And a reasonable value for our hydrostatic glomerular pressure can be something like 60 millimeters of mercury out, pushing the fluid out. But then our hydrostatic pressure pushing back in, we have to take into account, and that can be somewhere a good, I don't know, an average for that would be like 18 millimeters of mercury back in. And then our colloid osmotic pressure is going to be about 32 millimeters of mercury back in. And so our net filtration pressure then, if we take all of these things into consideration, is going to be about 10 millimeters of mercury out. And we want to keep our filtration rate pretty constant because you don't want to be changing your urine volume and blood volume on a moment-to-moment basis. You want to keep it in pretty tight homeostatic range. So we're going to look at that. This filtration rate, or the amount, the amount of filtrate that we produce in 1 minute, is what's called our glomerular filtration rate, and that's what we'll talk about next. So our glomerular filtration rate is our GFR. And we need to try and keep this pretty constant despite changes in systemic blood pressure, so that we're not changing blood volume on a moment-to-moment basis despite changes, small changes in blood pressure. So we've got to keep our GFR pretty constant so that we're making a homeostatic amount of urine and keeping our blood in homeostatic range.
Thank you.
Intrinsic controls go. These are our autoregulatory mechanisms, is what your book talks about. I think it names them autoregulation mechanisms. And we've got two. The first is the myogenic mechanism. Myo referring to muscle, genic being genesis. So the muscle is going to make the change in this type of control. And so that afferent arteriole has baroreceptors that are responsive to different levels of stretch. So if we get a drop in blood pressure, then that's sensed by the afferent arteriole. So this is sensed as a decrease in stretch. And the response then, if we didn't have a response, then the decrease in stretch would cause the afferent arteriole to constrict. And so if we're constricting, then we are going to drop basically lower the difference between the afferent and efferent arterioles. So you drop the hydrostatic pressure in there, you drop glomerular filtration rate. So we don't want that to happen. So what we're going to do is, with a decrease in blood pressure and a decrease in stretch, the afferent arteriole responds by dilating. And then when it dilates, now we can keep the hydrostatic pressure up despite the drop in blood pressure in the whole system. So this is going to increase that hydrostatic pressure in the glomerulus, which will increase our glomerular filtration rate so that we can stay constant. The inverse is also true. If we have an increase in blood pressure, then this is sensed as an increase in stretch. And if we were to just allow this to happen, then we would increase glomerular filtration rate beyond our normal homeostatic balance, and we don't want that to happen. So the afferent arteriole is going to respond by constricting. And then when it constricts, this will drop that hydrostatic pressure in the glomerulus and drop our glomerular filtration rate so that we can keep our glomerular filtration rate normal despite this external change in blood pressure. Any questions on that, on the myogenic mechanism? The second intrinsic control that we have occurs through this tubular glomerular feedback mechanism. And that video said that the juxtaglomerular apparatus is the afferent arteriole and the distal part of the nephron loop. That is the only book or site that I have seen say that. I checked every other anatomy book that I have in my house, and I have like 4, and they all say it's a distal convoluted tubule. So then I checked other websites. Harvard and Yale say it's a distal convoluted tubule. So So the tubular glomerular feedback mechanism is happening at the juxtaglomerular apparatus, which is that region where our distal convoluted tubule is contacting the afferent arteriole. And the macula densa cells in the distal convoluted tubule are sensitive to levels of sodium chloride passing through. So our macula densa cells are sensing sodium chloride levels. And if you've got a lot of sodium chloride in the filtrate passing through, then that's indicative that you're doing a lot of filtration. But we don't want to be doing a lot of filtration on a moment-to-moment basis. We want to do a normal amount of filtration. So if we have high sodium chloride, then this is saying that we have high GFR. So to come back to homeostasis then, we need these macula densa cells are going to use these other cells as kind of an in-between, go-between, they're called mesangial cells. And the mesangial cells are going to signal your JG cells to, in this case, constrict, so that we can drop the hydrostatic pressure in the glomerulus and drop our glomerular filtration rate. So if we have this increase in sodium, this is indicative of an increase in glomerular filtration rate, so we're going to respond by stimulating the JG cells to constrict. If they constrict, we decrease the diameter. We're going to decrease our hydrostatic pressure in the glomerulus, which will decrease our glomerular filtration rate and bring us back to homeostasis. The inverse is also true. If we've got low sodium chloride coming through, then this is indicative of low glomerular filtration rate. We're not filtering enough. The macula densa cells are going to sense this. Then they'll reach across through the mesangial cells and stimulate the afferent arteriole to dilate. And then if we dilate, then we have a bigger difference between our afferent and our afferent arteriole, so we'll increase our hydrostatic pressure in the glomerulus, which will increase our glomerular filtration rate and bring us back to homeostasis. Okay. As far as extrinsic control goes, our sympathetic nervous system under extreme stress is going to be able to override the intrinsic controls and stimulate vasoconstriction in that afferent arteriole. Then we're going to drop the hydrostatic pressure in the glomerulus, which is going to drop our glomerular filtration rate. If we drop our glomerular filtration rate, then ultimately you're going to drop the amount of urine that's produced. So that would increase our blood volume overall. The renin-angiotensin system is also going to have an effect because of vasoconstriction, but then also through aldosterone, and not at the juxtaglomerular apparatus, but in another place in the system, our antidiuretic hormone will have an effect. So if we have a decrease in blood pressure, this is going to be sensed by our JG cells, that are going to respond by releasing renin. We know that renin combines with renin substrate or angiotensinogen to become angiotensin 1. Angiotensin 1 goes to our lungs where it's converted by angiotensin converting enzyme to angiotensin 2. And angiotensin 2 is also a potent vasoconstrictor, so it will stimulate vasoconstriction at our afferent arteriole. So by stimulating this vasoconstriction, we're going to decrease our hydrostatic pressure in the glomerulus and decrease our glomerular filtration rate, ultimately then increasing the amount of blood that we have in the system, increasing our blood volume so that we can increase our blood pressure. The other thing angiotensin 2 is going to do is it's going to stimulate aldosterone release from our zona glomerulosa of the adrenal gland. That aldosterone then is going to stimulate the distal convoluted tubule to uptake sodium in the filtrate that's passing through, and water follows sodium. So this is going to stimulate our distal convoluted tubule to increase sodium reabsorption, and water passively follows. So that'll be another way that we can decrease our urine output and increase our blood volume. Angiotensin 2 is going to be able to have another effect in the system, not right there at the juxtaglomerular apparatus, but we might as well talk about it now. Angiotensin 2 also stimulates ADH release, or antidiuretic hormone. Antidiuretic hormone is going to stimulate our distal convoluted tubule and our collecting duct to uptake water or increase water reabsorption. So that's not happening right there at the juxtaglomerular apparatus per se. Further down in the distal convoluted tubule and throughout our collecting duct, ADH will have its effect. All right, so first we'll talk about tubular reabsorption, and then we'll talk about tubular secretion. So tubular reabsorption is how we're going to return useful materials from that filtrate back into the blood. So filtration doesn't care what's in your blood plasma. It's pushing everything out. It's a passive process that's pushing plasma and all of the dissolved solutes out. There's a lot of stuff in that you need. There's glucose, amino acids, water. We're going to need some ions. So we need to be able to pull that back, the useful things back out of that filtrate and bring it back into the blood. So tubular reabsorption is the process by which we do this. And it begins in the proximal convoluted tubule. So, and the proximal convoluted tubule actually does most of our reabsorption. And the things that it is reabsorbing, it's reabsorbing 100% of glucose. It's reabsorbing nutrients like amino acids, vitamins. Depending on what's going on in the system, what ions we need, it will also reabsorb ions. There's a whole process called urea or uric acid recycling. Your book addresses it a little bit. I'm not going to test you on it, but it's kind of weird because the proximal convoluted tubule can reabsorb urea and uric acid, but it can also secrete them. So it depends on what's going on in the system. The other things that can be absorbed would be urea or uric acid. And so that's mostly it. And again, the ions that we're reabsorbing is totally depending on what's going on in the body. So if we need more sodium, we'll bring in more sodium. If we need more potassium, we'll bring in more potassium. On a general day-to-day basis, potassium is something you want to excrete most of the time. You don't want a high extracellular level of potassium. So most of the time we're going to be secreting it, but sometimes you need to bring it back. So the proximal convoluted tubule can do that if need be. When we get to our nephron loop, the different limbs are going to have different things happening. So in our descending limb, water is allowed to leave. So water is being reabsorbed in the descending limb. And this is going to be important for forming an osmotic pressure gradient that we'll see for our collecting duct in just a little bit. So in the descending limb, we'll reabsorb water. And this is immediately taken up in the vasa recta. And that's important because it's not going to allow for our interstitial fluid to be dilute. When we round the hairpin turn and start up our ascending limb then, this is impermeable to water. So water gets stuck, but now we're going to reabsorb salts. So primarily sodium chloride, but you can also do some potassium reabsorption here. And this is going to be active transport out of the ascending limb and into the interstitial fluid. So it's active transport from our tubular fluid to the extracellular fluid. And that's going to help concentrate our extracellular fluid. We want to really have a concentrated extracellular fluid so that we can set up an osmotic pressure gradient to reclaim as much water as we can when we get to the collecting duct.
When you were saying, I'm sorry, the water was taken up in the vasa recta, you said it's important so it will not allow what to be dilute?
The extracellular fluid.
Okay.
So we're pumping all these salts out and we're leaving them in the extracellular fluid. Now you can take some of them up into the blood, but they have to be actively transported into the blood as well. So most of the salts are going to hang out in the extracellular fluid and help set up that osmotic pressure gradient. So it's like this whole nephron loop's job is to set up an osmotic pressure gradient for maximum water reabsorption. I'm going to show you another video that demonstrates that in just a minute because it makes this countercurrent multiplier make more sense. When we get to the distal convoluted tubule, everything that's happening in the distal convoluted tubule is under hormonal control. So this is under hormonal control. And we could say that aldosterone is stimulating sodium uptake or reabsorption. Water passively follows. Although if we have antidiuretic hormone present, then that's going to stimulate water uptake or water reabsorption. Other hormones that act on the system would be like parathyroid hormone. What do you think that's going to stimulate the uptake of? I heard somebody whisper it maybe.
I don't know what's hard. Calcium.
Calcium.
No.
So
Close.
parathyroid hormone is released in response to low calcium levels. So if we've got parathyroid hormone in the system, this is where it's targeting to increase that calcium reuptake. So it'll bring back calcium if it's out there in the system, and we need it. Then for our collecting ducts, they're also going to be under hormonal control of ADH. So ADH will help to stimulate water reabsorption. And the osmotic pressure gradient that we're setting up is going to have a huge effect on helping this out. So we'll see that in just a minute. Also, as part of the urea recycling, if we need to bring urea back into the body, if we don't have enough, if too much of it got secreted, then you can also reabsorb urea at the collecting ducts. The countercurrent mechanism has two parts, what's called the countercurrent multiplier, and that is set up because of what's happening in the loop of Henle. So the countercurrent multiplier is, as we're allowing water to flow out on our descending limb, that's going to concentrate the fluid in here, so it's becoming more concentrated. But then as we pass up into the ascending limb and that water can't get out and we're pumping out salts, it's going to redilute it and it's going to concentrate the extracellular fluid so that we get that osmotic pressure gradient. So that's the countercurrent multiplier. The countercurrent exchange is basically the vasa recta taking this water away to help create that osmotic pressure difference. So as our vasa recta pull this water away, that is responsible for helping to keep this extracellular fluid concentrated. So that we call the countercurrent exchange. And they're both countercurrent. In the loop of Henle, the current is running different from our fluid in the descending limb to the ascending limb. But then also in our vasa recta, the blood flow is flowing opposite of the fluid passing through the nephron loop, so it's also countercurrent. And we are pulling in this water then that's being excreted or being reabsorbed from the descending limb, and we're able to then concentrate our extracellular fluid because we're not actively pulling those salts back into the blood. So that's going to create that big osmotic pressure gradient that will allow for water reabsorption at the collecting duct. Any questions? Alright, as far as tubular secretion goes, remember tubular secretion is how we're going to remove unwanted materials from the blood and concentrate them in what's becoming urine. So we'll remove unwanted materials from blood to excrete them in the urine. So tubular secretion leads to excretion. And in the proximal convoluted tubule, it again is doing most of our secretion. So it is a lot longer and more convoluted than our distal convoluted tubule. So both of these processes are happening at the same time, but we've got a lot of time for that tubular fluid to pass through here. So our proximal convoluted tubule is doing most secretion. Things that we're going to secrete are things like our nitrogenous wastes, like ammonia. We'll also secrete urea and uric acid. We'll also secrete drugs. We'll secrete excess hormones. If your blood pH is off and where we're acidic, we can actually secrete hydrogen ions. This makes the kidneys a buffer system all on their own. As this tubular fluid enters the nephron loop in the descending limb, the only thing that we may secrete is urea. In the ascending limb, there's no significant secretion. To the distal convoluted tubule, one thing that I didn't mention is that when we're reuptaking or reabsorbing sodium, potassium's moving the opposite way. Those two ions always move the opposite way. So if we've got aldosterone present in the system stimulating sodium uptake, then we'll be excreting or secreting potassium. So with aldosterone present, we would secrete potassium. In this area, we can also secrete ammonia and hydrogen ions. And our collecting duct, as far as secretion goes, is going to be the same as our distal convoluted tubule. It's going to be able to secrete potassium, ammonia, and hydrogen ion. So most of our tubular secretion is happening in the proximal convoluted tubule.